Hello everyone. Today we are back with another video on DNA amplification through polymerase chain reaction, or PCR. So, let's get started. In order to make copies of pages you need a photostat machine, but to make millions of copies of DNA, we perform DNA amplification by different methods, one of which is polymerase chain reaction. Polymerase chain reaction is an in vitro method. In vitro means in the glass. Means that it is done outside of a living organism, and it usually involves isolated tissues, organs, or cells. For amplifying a selected DNA sequence that does not rely on the biologic cloning method, which is in vivo. In vivo means performed or taking place in a living organism. Polymerase chain reaction permits the synthesis of millions of copies of a specific nucleotide sequence in a few hours. It can amplify the sequence even when the targeted sequence makes up less than 1 part in a million of the total initial sample. Gene amplification refers to an increase in the number of copies of a gene in a genome. The method can be used to amplify DNA sequences from any source, including viral, bacterial, plant, or animal. Procedure for polymerase chain reaction uses DNA polymerase to repetitively amplify targeted portions of genomic or complementary DNA. Each cycle of amplification doubles the amount of DNA in the sample, leading to an exponential increase. Exponential increase means 2n, where n is the cycle number in amplification. The amplified deoxyribonucleic acid products can then be separated by gel electrophoresis. Gel electrophoresis is a laboratory method used to separate mixtures of deoxyribonucleic acid, ribonucleic acid or proteins, according to molecular size. In gel electrophoresis, the molecules to be separated are pushed by an electrical field through a gel that contains small pores, while detected by southern blotting. Southern blotting is a laboratory technique used to detect a specific DNA sequence in a blood or tissue sample. And hybridization. Hybridization as related to genomics. It is the process in which 2 complementary single-stranded deoxyribonucleic acid or ribonucleic acid molecules bond together to form a double-stranded molecule. The bonding is dependent on the appropriate base pairing across the 2 single-stranded molecules and sequenced. Now let's discuss the basic concepts of DNA amplification through polymerase chain reaction. First is the constructing primer. Well, it is not necessary to know the nucleotide sequence of the target DNA in the polymerase chain reaction method. However, it is necessary to know the nucleotide sequence of short segments on each side of the target DNA. These stretches, called flanking sequences, they bracket the DNA sequence of interest. The nucleotide sequences of the flanking regions are used to construct 2 single-stranded oligonucleotides, usually 20 to 35 nucleotides long, which are complementary to the respective flanking sequences. The 3' hydroxyl end of each oligonucleotide points toward the target sequence. These synthetic oligonucleotides function as primers in polymerase chain reaction. Moving towards sample preparation. Well, a sample for analysis is prepared by adding the deoxyribonucleic acid, either genomic deoxyribonucleic acid or complementary deoxyribonucleic acid, the primers, an excess of deoxynucleoside triphosphate, and a heat-stable deoxyribonucleic acid polymerase, to an appropriate buffer solution. It was all about this video on this topic. In our coming section, we will discuss about the steps of polymerase chain reaction.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.